my name is Hiran Patel. I'm a pediatric hematology oncology fellow. I work at Cohen's Children's Medical Center, and I conduct my research at the Feinstein Institute of Medical Research. I'm going to be speaking about my project looking at RPS19 haploinsufficiency in the skeletal formation in DBA. The objectives of my talk: first, I'll go through an overview of Diamond-Blackfan anemia, including the clinical and genetic features of this syndrome. Next, I'll describe our work on the skeletal phenotype of DBA. And finally, I'll discuss a model that we're going to use to study osteosarcoma. Diamond-Blackfan anemia is an inherited bone marrow failure syndrome. It's a rare disorder. It affects between 1 in 100 to 200,000 individuals. Phenotypically, it presents with erythroid hypoplasia. Typically, patients are identified in early life with evidence of erythroid failure such as macrocytic anemia and reticulocytopenia. In addition, patients have short stature, congenital anomalies like heart defects, and DBA is a known cancer predisposition syndrome. Most commonly, mutations that cause DBA are dominantly inherited, leading to ribosomal protein haploinsufficiency. The mutations affect protein components of the small and large ribosome subunits, as you see there on the right. In the literature, much work has been done defining the mechanisms of erythroid failure. Much less work has looked into the skeletal phenotype. In the past, the short stature present in DBA was attributed to chronic anemia and corticosteroid use. However, there's more to the story, and this is the focus of our research. We use a mouse embryonic stem cell line harboring a RPS19 gene trap to model osteoblast differentiation. This model allowed us to model RPS19 haploinsufficiency. Using alizarin red staining, we show that by day 12, wild-type cells show robust mineralization, whereas RPS19 haploinsufficient cells show defective mineralization that persists all the way to day 23. To understand the mechanism of decreased osteoblast activity, we perform gene expression studies. We observe that there's dysregulation of osteogenesis, with the exception of osteoclast function. Given the findings in our in vitro data, we look to see if the effects of RPS19 haploinsufficiency is conserved in the native bone environment by developing a mouse model. We used CRISPR-Cas9 to insert LOXP sites flanking exon 2 of the RPS19 gene. These mice were bred with mice with a PRRX1 Cre transgene to allow for a conditional deletion of RPS19 in the early limb bud mesenchyme. What we see are impressive skeletal findings. In our wild-type mice, you can see there that they're normal in size, whereas our RPS19 haploinsufficient mice are smaller. In addition, they show forelimb and hindlimb defects. Using alizarin red and alizarin blue staining, the calvaria show that the sutures and the fontanelles are widened in RPS19 haploinsufficient mice. In addition, looking at the forelimbs, the humerus is smaller in size. There's an absence of the deltoid tuberosity. In addition, there's agenesis of the scapula. And these findings persist in 10-week-old mice. By tunnel assay, we demonstrate increased apoptosis in the mouse limb growth plate. In the literature, this is in part mediated by the Wnt pathway. So we did gene expression studies. And we show that downregulation of the Wnt pathway is involved in this model. Given these findings, we wanted to understand the role of P53 in mediating these defects. We chose P53 as extensive work in the red cell compartment of DBA shows apoptosis mediated by P53 activation. Mechanistically, ribosomal protein haploinsufficiency leads to a loss of P53 degradation via MDM2 inhibition. In our mouse model, we observe increased p53 in the growth plate of haploinsufficient mice. Having established p53 effects in RPS19 haploinsufficient mice, we next mated our mice with p53 conditional mice to observe the effect of p53 deletion in vivo. And what we observe is a dose-dependent rescue of the skeletal abnormalities. So as you can see here, as we delete one and then two alleles of p53, the fontanelles and the sutures normalize. In addition, the limbs increase in size, and we see less defects in the scapula. Our findings were recapitulated in a human model of RPS19 haploinsufficiency. Here, we show similar defects in a stillborn fetus with heterozygous RPS19 deletion. In this fetus with RPS19 haploinsufficiency, there's a decrease in gestational size. In addition, the skull is the size of an infant at 34 weeks of age. Histologically, the growth plates are disorganized as we see in our mouse model. Together, this shows the translational potential of our work. The final part of the story of DBA is its role as a cancer predisposition syndrome. Work done by our Diamond-Blackfan anemia registry demonstrated that there is a strong association with cancer formation, specifically osteosarcoma. Preliminarily, in our mouse model, we've seen osteosarcoma development in 4 mice between the ages of 10 and 28 weeks, which is a young age for tumor formation. This is in the background of a single allele deletion of RPS19 and TP53. What we hypothesize is that RP haploinsufficiency leads to nucleolar stress. This selective pressure results in the selection of cells with loss of function of TP53, resulting in osteosarcoma. This model shows potential towards expanding our knowledge of cancer predisposition in DBA, and in addition, studying therapeutic options to ameliorate this process. So to summarize, we spoke about an overview of the clinical aspects of DBA. In addition, we showed that RPS19 haploinsufficiency leads to defective osteoblast differentiation in vitro. In vivo, our mouse model of RPS19 haploinsufficiency recapitulates the skeletal defects seen in humans, and this is in part mediated by p53. And preliminarily, osteosarcoma develops in mice with a single deletion of RPS19 and p53. As far as future directions, we're going to continue expanding our work on the mechanisms of mesenchymal failure in DBA. In addition, we're going to be launching our osteosarcoma research using DBA as a model. Thank you, and I'm open to answer any questions.